Good evening. We'd like to welcome you to our St. Luke's lecture, which is something that we typically put on annually through Sacred Heart Mercy Healthcare Center, which is an apostolic work of our community of the Religious Sisters of Mercy. We especially want to welcome our online audience tonight. For those who are attending and hope to gain continued medical education credits or social work credits, if you've registered for the link with our clinic, that information will be emailed to you. If you didn't pre-register, the QR code and the information will be available at the end of this presentation, and it'll be on sequential pages, so just watch for that. We are very pleased to welcome Dr. William Hurlbut. We've been trying to get Dr. Hurlbut here for about 2 years, but with the pandemic, we needed to delay this event, and so I'm sure it's been worth the wait. His coming is particularly poignant at this time, given the threats to human life that are being proposed in the state of Michigan. Dr. Hurlbut has had a vast professional career and is continuing to do so as an educator in medicine and ethics, author, and as an expert witness in many areas of government. And I will just highlight a few notes from his impressive, extensive biography. Dr. Hurlbut is a physician and adjunct professor in the Department of Neurobiology at Stanford University Medical Center. In addition to his medical training at Stanford, he has completed postdoctoral studies in both theology and medical ethics. His primary areas of interest involve the ethical issues associated with advancing biomedical technology, the biological basis of moral awareness, and studies in the integration of theology and philosophy of biology. He was instrumental in establishing the first course in biomedical ethics at Stanford Medical Center. Dr. Hurlbut is the author of numerous publications on science and ethics. He has organized and co-chaired 3 multi-year interdisciplinary faculty projects at Stanford University. Dr. Hurlbut has provided expert testimony to national and international governments, as well as serving on the President's Council on Bioethics from 2002 to 2009. Please help me welcome Dr. Hurlbut.
Thank you, sister. It's really wonderful to be here with people who are I'm learning so much and enjoying so much being with you. So I want to talk with you about a subject that is, to my mind, of very great importance in our current era. And month to month, including within the last couple of weeks, some of the news that's come out make it all the more powerful and important. What I want to do is I want to explain to you how in our hearing, in our lifetime, we're moving from an era of discovery in molecular biology, and it culminated in genetics. Now we're moving on to the era of developmental biology, where we study what all those genes actually do to knit together a living human organism. And I want to do this by describing the new tool called CRISPR-Cas9, which is a form of gene editing that is revolutionizing advances in biotechnology. And it holds tremendous power for good. And I want to say that right off so that nobody gets confused that I'm negative about this, because I think it's a little like Moses on the mountain. Today, I give you a blessing or a curse. We have a great opportunity to do good. And I think some of this can come right through the medical center here if we handle it right. If we don't handle it right, we're looking at nightmares that will approach some of the worst in history. And so what I want to do is show you how this new tool of genetic engineering allows us to learn more about the nature of our human organism, of our being, and what challenges it poses. And then I want to show you what some of the ethical minefields are in it and what some of the ethical opportunities are in it. So that's my basic agenda. And I thought I would begin by showing you a slide from a conference that I gave a talk at Harvard some years before COVID now. Actually, I've been there for a while, but it's called Editorial Aspirations. And I thought it was a very interesting poster because it highlights the human hands, what Aristotle called the tool of tools, the symbol of our unique body form and capacities of mind, our creativity, our comprehensive operation and control over the world in which we are. And nowhere is it more dramatic what those hands are doing than in the ongoing unfolding of biotechnology. Those hands are turning now to operate on our very selves. So we're at the beginning of an amazing new era of opportunity, new era of genomic editing. MIT Tech Review called this the biggest biotech discovery of the century. And it's commonly known as CRISPR. And if you're not familiar with it, it's just an acronym based on the way it was derived. Its discovery was based on a kind of immune system where bacteria protect themselves against viruses by cutting up the virus. And this clever scientist, Jennifer Doudna, figured out that the same mechanism could be used to edit genes. And it's been called a molecular scissors in that regard. But it's actually more rightly considered to be like a Swiss Army knife because it can do such a variety of unique things in operating on the human genome or any other living genome for that matter. What it amounts to is a tool whereby one part, a little RNA segment, identifies a DNA segment. You remember from high school, they're complementary, high school biology. And so it locates a specific part of the DNA, your chromosomes, and then it cuts. It's got another little portion called an endonuclease that can cut, but that little cutting mechanism can do a whole variety of operations on the genome. So it's an amazingly versatile tool for operations on every level of genomic process. And it advances gene editing to the level where Jennifer Doudna, the discoverer of CRISPR-Cas9, has declared that that we're able to rewrite the very, did I do something bad? No, I'm having trouble moving my slides, so I'm moving on up here. Is that okay? Did I do anything wrong? I unplugged we can plug that back in when I do the video, okay? Okay, so Jennifer Doudna says, advances in gene editing are enabling us to rewrite the very language of life. And I think that's not an overstatement. It's really amazing. What can we do? We can make more nutritious fruit. We can make plants that make photosynthesis more efficient and therefore can produce things like biofuels. We can alter plants or animals to add in human genes to make proteins that can then serve various biological functions, including anti-cancer drugs. We can modify animals so that they can then produce organs that can be transplanted into human beings. Essentially, this is a tool for knocking out or knocking in any kind of gene, pretty much any kind of gene. It's got some limitations, but they're rapidly being transcended. But the most important part of this conceptually and for biomedicine is its operation on very early human development. Now, as I said at the beginning, we spent the 20th century doing biology in a test tube, basically, learning about the ingredients. Now we're understanding what those ingredients do. And we all know this because in the early part of the century, we all were excited by and also troubled by this phenomenon known as embryonic stem cells. The idea was if you could take those early cells, you could coax them to become any kind of cell in the body and therefore both discover how the body is knit together, but also produce tools that would allow therapeutic intervention with cell therapies. And although this is going slowly, it is slowly but steadily giving us new insights and operations in biomedicine. So unlike other previous gene editing methods, which by the way were introduced while I was a med student at Stanford, one of my biochemistry teachers got the Nobel Prize for that. It's Paul Berg. But unlike the succeeding decades since then, when we haven't had a really efficient tool, this new tool is cheap, quick, easy to use, and it's swept through labs around the world. There are literally thousands of serious publications using this technology. Just to give you an idea how powerful this is, I know the guy who did made the first gene-edited mammal, a mouse. He did one gene. He told me that it took 2 years, $200,000, and he could change just one gene. I had dinner with him just before COVID, and he said, now for $2,000 in 3 weeks, I can change dozens of genes. So you can see immediately how powerful that is, how much advance in theoretical understanding and practical intervention it allows. And this has this has been a quantum leap in advances in basic techniques of biotechnology. And we're right on the early edge of a revolution in technology that equals and will transcend earlier revolutions in synthetic chemistry and physics. So the end of the 19th century, they had the periodic table of elements, and you know what's come from that. Chemistry has just remade our world practically, and physics, of course, too. Now we're entering the vast realm of biology, and it touches everything associated with life and will be used for things that aren't necessarily directly associated with life. Believe it or not, you can actually encode the entire Library of Congress in a very small amount of DNA. So there are really amazing possibilities here. So Jennifer went on to say, we're standing on the cusp of a new era, one in which we will have primary authority over life's genetic makeup and all its vibrant and varied outputs. So what will we do? Well, earlier scientists had used gene editing to put firefly genes in mice so they'd glow in the dark. That's funny, probably didn't hurt the animal. Somebody made a bunny that also glowed in the dark for an art project. Scientists have created albino lizards, taking the pigment cell out, and that probably doesn't hurt the animals, and it creates interesting opportunities to study development. In our era, I think the possibilities, we barely begin to think of them. But among other things, the mating barriers between species are based on very simple things, a few proteins that prevent the sperm from penetrating the egg. And in a certain number of cases, there will be developmental possibilities that will go on to birth. So I think it's possible by influencing what can mate with what and how the genes actually express themselves during development, we may see a whole new kind of biotechnological zoo. And Dr. Zeus may be out of fashion right now. He's been canceled for reading to kids, but we may take them to see the Dr. Zeus Zoo. I think this is really going to happen because there'll be fascination and commercial interest in it. That's a serious matter. We got to make sure we don't create creatures that are suffering. And so that's a side issue. But then, of course, when it comes to hybridizations and chimerizations of cells, mixing and matching and reorganizing biology will manifest itself in very interesting possibilities for human-animal chimeras, such as something like this. Now, don't worry, this is just a drawing. It doesn't exist. But I think we will see some very strange combinations of human and animal, and we have to gird ourselves up ethically against things that are a violation of human dignity. Nonetheless, there are some very useful tools to be gained in perspectives in biomedicine through the chimerization of human organs, for example, developed in other animals, and studies of the way genes operate and the way cells migrate and so forth. So I'm not utterly against it. It's just that we need to frame it up with intelligent considerations. Already scientists in China have put human genes into monkeys to make them so smarter. They could do some things better, but they could do other things not as well. And this has a long history in science or pseudoscience. 100 years ago, Stalin wanted to create the human Z, and he had a whole agenda for why he wanted to do this, including making soldiers that wouldn't care what they ate and would, be able to resist pain better. Just the kind of people Putin would like to send to the Ukraine today. But fortunately, it didn't work out very well. But in our own era, very prominent scientists, including a guy I know pretty well at Harvard, George Church, has spoken of the possibility of de-extinctioning some of our human ancestors, like Neanderthals and Denisovans. So the question arises, if we did that, we'd learn a little bit about what made human beings unique, but what would we do with these creatures? Would we consider them human? Would we allow them to mate? Would they mate with each other? Would we allow them to mate with humans? We do have a history, by the way, in our species of having mated with Neanderthals, and they say the average human being carries about 3% Neanderthal genes. So this is a very fascinating subject, but obviously full of controversy. And another controversy would be if we intend to leave this Earth behind and go out into space. Well, if we're going to do that, we're going to be subject to all sorts of physical forces for which we're not equipped by our nature. So genetic engineering of human beings may arguably be justified if that's an imperative of extension of our species, but it's very iffy. I mean, how much risk are you going to take to make a human being adapt to space? I think it's going to be very challenging, very troubling, actually. I'm not, by the way, negative about it exactly. I think it's exciting in some sense, just like sending off Magellan was exciting, but I think it's going to be far more dangerous. Although I wasn't there with Magellan, who knows? But anyway, so astronauts, and then there are all sorts of other ideas here. There's a joke, I'm sure, but somebody suggested we just create very, very small human beings so they wouldn't use up so many of the resources of the Earth. Just think how many of them could live right here in Alma if they were just 1/5 the size. One of my colleagues on the President's Council on Bioethics, Francis Fukuyama, wrote a book called Our Post-Human Future, and there's quite a whole culture now evolving of people that they are excited about the whole idea of techno-sapiens, machine animals, hybrid hybrids, and so forth. And they have a whole agenda, and the key among them is to create immortal creatures. That's obviously a pipe dream, but there is a huge amount of energy and resource being poured into longevity studies, including Google has a billion-dollar company called Calico, California Life Institute. And Jeff Bezos has funded something called, I think it's called Altos Lab, and he's luring Stanford professors further down Silicon Valley by giving them salaries of $1 million a year. And they hope to crack the nut of aging. Well, it raises a strange question. Is aging a disease, or is it a natural process of human life? And is it even a good process of human life whereby we live a life cycle that has meaning and is generative and then makes room for the next generation? I'm actually of the mind of that latter statement. I think I'm okay with getting older and dying, at least while I'm not too close to it. Who knows how I'll feel as I get closer, but I think a compassionate and thoughtful and spiritually sound view understands life as a life cycle. This is a famous statement by one of the leading scientists at the time. The real goal is to keep people alive forever. And that's expressed in this new cultural movement called the transhumanism. And the idea is that we are going to transcend the cruelty and contingency of our origins and take hold of our human future and alter, using genetic engineering and a variety of other technologies, altering ourselves both physically and thereby culturally in capacity to transcend. Everybody knows what this means who's been to school. It's H+. So these ideas have been taken up by the very graceful and thoughtful writer Yuval Noah Harari. I'm betting that. How many of you have read Sapiens or Homo Deus? Anybody in the well, that's real. I'm very happy to see you haven't done that, because I read both those books because they're so popular. Highly recommended by Bill Gates and Mark Zuckerberg, who started Facebook and touted as a great intellectual. I just don't agree. I think he's way out of his depth when he talks about these matters. I'd love to debate him. He says sapiens will upgrade itself into another kind of being within a couple of centuries at most. Earth will be populated by beings who are different from us in their cognitive and physical abilities. If I had to guess, I'd say 200 years from now, we're going to be pretty much the same species we are today. Now, if we get challenged by something like a virus, like COVID, only very more serious, that we can't transcend, maybe we will engineer it ourselves, and maybe we'll end up engineering away in this human lineage some of these terrible diseases I'll tell you about in a minute. But in terms of improving human beings, we don't know nearly enough, and I'm not sure we'll ever know enough. And then when we do know enough, we may appreciate why we are the way we are. So I'm just guessing, and I may be wrong. There was a whole history of Christian reflection on this, including what's called Russian cosmism, where the belief was that human beings were intended by God to be the agents of their own creation and to bring about a kind of improved utopian reality. My own feeling is that these tools should be used in a compassionate way and that they should be guided by the principle of healing, not by transcendent physical rearrangements. But that's a subject of great debate and discussion, and I'm not the final word in it. However, I have thought about it quite a bit, and I'll tell you why I don't think it's going to happen. But in the meantime, before I do that, I want to show you what it can be good for, uncontroversially, I think, or pretty nearly uncontroversially. Jennifer Doudna goes on to say, we may be nearing the beginning of the end of genetic disease. What a statement. Imagine there are about, in just single gene diseases alone, there are at least 6,000 to 8,000. There's probably tens of thousands we haven't discovered yet. And imagine if we could cure these diseases. By the way, I know Jennifer Doudna very well. She got the Nobel Prize a year ago, practically to the day. And I did a 4-year project with her on the ethics of this. So I'll tell you more about that later. But she's quite a thoughtful person, a good person in spirit, I'd say. When it comes to scientific perspective, she has a very deep conscience about this, although she admits to everybody that she doesn't understand these ethical issues in their depth, and she's leaving it up to us who are more theologically and philosophically trained to think about it. But she also is encouraging that process to take place, so I respect her for that. So what are some of the possibilities directly, immediately, already happening? Well, the primary one that's most prominent, you've probably seen it in the news, is sickle cell anemia. That's caused by a single base pair. That's a single little unit of DNA that is mutated. And it's not randomly mutated in the sense that it's irrelevant, because as most of you know, it turns out that sickle cell anemia, when found in just one of the parents producing the baby is protected somewhat against malaria. So it has a reason for its being in the human genome. But when you get it from both of your parents, you have sickle cell disease, and then you're subject to a very debilitating disorder. And you see the little sickles there in the blood smear? This is the way blood's supposed to look. It's the little smooth biconcave discs that flow through even the smallest little capillaries very smoothly. But these sickles, they clump, they form clots when they're in an environment without enough oxygen. So if you get an injury that's slowing blood flow or some other cause of lower oxygen levels in the blood vessels, you get a little clot and a little clump. And then that just expands until it's blocking off a larger piece of tissue, and that causes great pain. And you go into what's called sickle crisis. I saw a little 11-year-old African-American boy at Stanford Hospital when I was in training, who was in for his 200th hospitalization. A poor little guy who was suffering, and it would be wonderful if we could cure him. Well, the way scientists do this is they mobilize cells from the bloodstream, they take them out into the dish, and they modify them genetically and put them back in, and they repopulate the marrow. They're the special cells that are called hematopoietic stem cells that can repopulate and produce healthy blood cells. And there's a variety of ways to do this, but you get the point. Genetic engineering can cure this disease. And it looks like there are some patients in our world today who've been cured already. It's experimental, but it's getting there. And for tissues that are hard to reach, you can't just remove cells. We have viruses that can be programmed to specific addresses. It's fantastic. I mean, it's viruses called, well, there are a variety of kinds, but adeno-associated viruses can be engineered to go specifically to just one type of tissue, including, by the way, even into inner tissues in the brain. So we may one day be able to modify the specific pathological problems in certain diseases that plague humanity. And there are many of them, as I said a minute ago, perhaps tens of thousands. This poor little fellow has something called Miller's syndrome. This little guy has something called Creutzfeldt-Jakob disease. And it's a very tragic disease. It's a metabolic disease. And these kids actually chew their fingers off and chew their lips off. And so if we could cure this, it'd be very, very wonderful. But what about this? What is this? Is this a disorder? Is this a disease? This is albinism. Do we want to, how much risk would we take to remediate this lack of pigment like the salamanders I showed you? Albinos live good, normal human lives. And they have to be careful not to be in the sun too much, and they suffer a little bit of social exclusion. Believe it or not, in Africa, there's a demonic theory operating that if you can eat part of an albino, you can actually be cured of a whole host of diseases. So albinos are actually being captured and butchered for their parts in places like Uganda and East and Central Africa. So it's really, boy, it's not evidence of demonic forces, I'd say. But what about benign uses, completely more elective uses? Should parents be able to choose the eye color, hair color, height of their kids? Well, it turns out that partly we're going to be rescued by the difficulty of these agendas. It turns out that genes are not like, it's not like Mr. Potato Head or Legos where you just sort of plop a new thing on and it's not like there's a gene for the nose or the ear. Genes produce proteins. You remember that DNA produces RNA, which then produces proteins, remember? And so what happens is the primary proteins then go out and they mix with other proteins and operate in the whole context of the organism. So proteins are really more like the primary colors on an artist's palette, and they're all mixed before they actually operate. And there are very few things where a single gene can produce a healthy trait, many where they can produce abnormalities. And usually those are multiple, by the way, like the little Miller's syndrome I showed you. He's got multiple problems in his body. And it turns out that if you learn two words from me tonight, pleiotropy and polygenic inheritance. Polygenic inheritance means that most traits are caused by many genes. And it turns out that even something as simple as height is probably influenced by hundreds of genes. And all the more something like intelligence or longevity. It's not going to be easy to do anything we really want to do, like create more beautiful, intelligent, long-lived human beings. It's just not as simple as that. We may make a little bit of difference in some dimension if we did it in an unbridled way, but that's polygenic inheritance, and pleiotropy means that every gene does many things. So even if you want to target one thing, you'd get a lot of associated changes too in other systems that you didn't necessarily want to change. So the organism is a vast web of interconnected parts that works as an integrated unit. And it's that mysterious principle of unity that characterizes an embryonic being from conception on, by the way. So intelligence, beauty, not easy. If you wanted red hair, we probably could do that. Appears to be caused by They used to think it was just one gene, but maybe they think now it's a few. But we could probably give you that if you're willing to take the risks and see. But what about other things? Well, this is George Church. I mentioned him earlier. George Church is a geneticist at Harvard, and he has identified what he calls rare protective variants of large impact. And these are traits that exist in a certain number of people, but only in a relatively few people. And he's got a list of these, and this is about 5 years old, so he's probably got twice as many now. But he thinks there are certain genes that are identified with definite advantages for the people who have them. Extra strong bones, resistance to virus, sensitivity to pain, I'm not sure what low odor production is all about, but anyway, whatever. So he's got a whole list of them. And you probably saw in the last couple of weeks, there have been articles that certain people don't seem to be vulnerable at all to COVID. And I'm hoping I'm one of them. I haven't had it yet. But this is because there are gene variants that, see, what it turns out is biology is a great balance. An organism has to deal with so many things, not just the thousands of potential parasites and microorganisms that are trying to eat us, but all the challenges of temperature regulation, food resources, the mental operations that keep us going. And so biology is a great balance. I taught a course with a Nobel laureate named Baruch Blumberg, who discovered hepatitis B. And he used to say to our students, there are no good genes and bad genes in a healthy person. There are genes that balance off other things and different emphases of traits. And that's why we are all benevolently and beautifully unique. So that's a very positive thing, but none of us can have every advantage or every power. We need each other to complement our strengths and compensate for our weaknesses. So even this may not be as easy as stated, because there are a lot of associated questions. What else is going to change if you change these genes? But maybe but these are pretty minor things relative to the large scale organismal integration. And besides, as developmental biologist Stuart Newman points out, even if you start doing any of this, it opens the door to what constitutes a definition of normal and what constitutes an acceptable risk. So there's problems associated with this. Not only that, but the question of what constitutes a disease at all. You can say, well, we'll only do interventions that are toward disease, but what counts as a disease? So this, believe it or not, was a textbook defined disease in the antebellum South. And it was called drapetomania. I got corrected last week. Some African-American guy told me it's drapetomania. But anyway, it means from the Greek words meaning a kind of passion or intense desire to run away. And you can see the treatment was whipping. And that was in our medical textbooks. Isn't that shameful? And of course, you all know what the Nazis did, defining some people as vermin. And we know, I won't dwell on this, but we know how that ended. So what about it? Well, you might say, well, okay, those are extremes, but there are good things we could do. And anyway, it's pretty apparent that human beings have a huge appetite for improving their existence. So the Roman physician, 2nd century Roman physician, Galen, said, the physician is only nature's assistant. But look at what medicine's become in our own era. Even when I was a medical student, this was just beginning, but now it's very prominent. Tens of billions of dollars go into this kind of medical intervention for human improvement. And who doesn't want to be kind of perfect or whatever perfect is? Look at what we've got already. Rogaine, it treats male pattern baldness. When I was trained as a med student at Stanford, male pattern baldness was not considered a disease. And I'm glad we're sort of accepting baldness now. It's even kind of athletic and manly, but there was a time when losing your hair was sort of an embarrassment to people, and still is to some people. So they, look at this genius Madison Avenue people, they advertised Rogaine, one of the early drugs. And it was brought forward through the benevolent offices of the medical profession. And it says, this is Madison Avenue advertising genius. If you're concerned about hair loss, see your doctor. And in the small print, it says, if you're losing your hair, you no longer have a reason to lose hope. Can you imagine? But look at it. There he is on the windswept beach, late autumn, and he's got that thinning hair on the top, and he's feeling like it's the waning of his sexual prowess and his life happiness, and he's not going to be attractive. And I mean, it's just how to make you feel bad about yourself. And of course, we already have growth hormone, and I see there's a reason by some perspectives to help short people get taller because, it's a crazy thing, but we care a lot about those things. But we have so many other interventions ranging from birth control pills to Viagra to a whole range of things. And you can just tell where it's going to go. By the way, if you're interested in this subject, the President's Council, on which I served, produced I think it's the best thing we did, a volume called Beyond Therapy, Biotechnology in the Pursuit of Happiness. And it's a thoughtful perspective on these kinds of interventions. But just think where people would go. I mean, who doesn't want a healthy, happy baby of high potential? And what would we do to alter and intervene toward better brains? It's coming. I mean, whether it be gene interventions or direct protein, small molecule targets of development or operation of our physiological systems, including our mental operations, it's coming. It's going to be hard to resist. It's no longer sci-fi. People are willing to use interventions on a medical paradigm based on the original idea of cosmetic surgery. Now we're going on to lifestyle interventions, a kind of spirit of technological transcendence, interventions in the interest of anything that can overcome the imperfect, inconvenient, or unattractive. So let's be aware of that medicine's strange new role. It's no longer sci-fi, but it's very kind of mathematical. It's cold. It treats people as though they are machines and is not necessarily sensitive to the very kind of diversity and what some people call the messiness of the blessed parts of human existence. So in light of all this, Jennifer Doudna started having nightmares about where her discoveries could go, and she helped to organize the first human gene editing summit by the National Academies of Science. This took place in December 2015, but just a few months after it had been scheduled, before it took place, the Chinese announced they had genetically modified human embryos, albeit embryos that seemed to have a defect that couldn't let them develop much farther than early embryonic stages. Meanwhile, scientists such as my own professor, Paul Berg, Nobel laureate who discovered genetic engineering, had said things like, to address complex diseases like cancer, we must carry our investigations to the most fundamental elements of living systems. That means operating, genetically altering, and so forth, human embryos. And George Daly, Dean of Harvard Medical School, says gene editing could be used to engineer specific disease-related mutations in an embryo, which could then be used to produce embryonic stem cells that could act as models for testing drugs and other interventions in disease. And if there were no ethical issues, I would wholeheartedly agree with this. But the question is, what is a human embryo? Is it really that ambiguous? It's a living human organism. It's the beginning stage of human life. And people are confused about this. They call it all sorts of things like a clump of cells and so forth. But that clump of cells is not like any clump of cells you'll find in a sedition in a laboratory. That clump of cells, if you will, if you want to call it a clump descriptively, it contains within itself the power of its own development. In biology, the whole, as the unified principle of life, precedes and produces its parts. It's not like an assembly line where you don't have a car until you put the steering wheel on. It's self-developing. So with all that in mind, the controversies, they knew they had to tiptoe around the controversies, because anybody thoughtful knows that the embryonic stem cell debate, which took place during the years I was on the President's Council, so I had a front row seat on that. It was very bitter. You all know that. Well, that was just mere prelude to what's coming now. We're really looking at serious interventions and massive uses of human embryos potentially, not the least of which would be for genetically engineering out diseases. So if you're going to actually do germline genetic engineering, replace a gene so that the whole lineage of the future of a family won't have that disease, which sounds like a laudable goal, but if you want to do that, you're going to have to study it on a whole bunch of embryos so that you don't make an error in the one you're going to bring to life and produce a life that's very full of suffering. So if there were no moral issues, yes, we'd do that. But how many embryos would we use to figure it out? And so there was considerable anguish about this and considerable caution, even among the National Academy. They called for very great caution before going into this, and also for general consensus in the society so that it was both safe and accepted. And of course, the general goal was the belief that we could eventually get to that point. Well, at one of the conferences that Jennifer Doudna and I organized, we invited a young scientist from China named He, otherwise known as JK, because that's his name's too hard to pronounce, so everybody calls him JK. And he came and he talked to us a little bit, 10 minutes, about his work on mouse embryos and stuff. And then about 3 months later, I got an email from him saying, I'm coming through Stanford. Could I come and talk to you? So JK came and I said, yeah, let's have lunch. We'll meet outside the student union. And so I met him, and I thought I'd just spend an hour with him and go back to my office. I didn't have anything on my schedule that afternoon. I just talked with him and encourage him a little and go back. Well, I ended up talking with almost the whole afternoon. And the very first thing he said to me was, Dr. Hurlbut, what is going on in America that people are concerned about embryos? Is it just like a splinter, a fringe of people, kind of fanatical people who are worried about that? And I said, oh no, JK, it's at least half the society feels like that's a moral breach. And even those who favor overriding that concern may have some concerns about the embryo, but they think the science is so important that it justifies it. So no, it's a serious issue. And I remember very distinctly, the first question he asked me after that one was, how can something this small be as important as my little 2-year-old daughter if she got sick? And I said to him, JK, your 2-year-old daughter was once that small. And that's the point. So JK kept coming back to see me and talking 3 or 4 times. We had long conversations, and I was worried that he was getting more and more inclined to advance the science faster than was justified. At one point, I admonished him strongly. I said, JK, you better be careful, slow down. I had no idea what he was doing, but I just could feel it. Be careful, JK, don't do this. You're going to humiliate yourself, your family, your country, and you're going to humiliate science as a whole, as a global enterprise. And he just listened, but I guess it was too late, because at the second International Summit on Gene Editing, I was on my way to Hong Kong. This was in late November 2018. And I got a call from Antonio Regalado at MIT Tech Review and said, what do you know about JK? And I said, oh no, don't tell me. JK's implanted embryos. And he said, I think there are born babies. So I bought the Wi-Fi all the way across the Pacific, and in typical form, the Wi-Fi didn't work. So $34, and it didn't work. But finally, an hour out of Hong Kong, it clicked in, and every headline in every newspaper of the world was genetically modified twins. And you all remember this. It was a big enough deal in sort of mainstream America, but you should have felt what it was like at this gene editing summit. People couldn't believe it. Those kind of summits are organized partly as a public relations thing that says, hey, we're the scientists, we know their problems and we can handle them. But this was like hiring a skywriter saying we can't handle them. And it was a big fiasco. And JK was supposed to talk just about his little science experiments, but by the second day when he was supposed to talk, they'd cleared an hour and a half. And he was brought in with an armed guard, and he gave his defense of his science and what he'd done. I knew him so well, I was supposed to go have dinner with his family the night after his presentation. Of course, that never happened. But a couple of days after I got back from Hong Kong, I got an email from JK, and this is a picture of JK. Nice, he's actually a very nice person and a very idealistic person. There he is in my dining room in California. And what he did was he altered a gene that is a sort of a portal, a receptor gene that allows the HIV virus to get in, and therefore the person who gets this editing would theoretically not be vulnerable to HIV, AIDS. So he had a good intention, and I can tell you more about why that was, to his mind, a good intention later. But he opened up a door that should not have been opened up, definitely not at that time, and perhaps never. I had dinner with Francis Collins, former head of the NIH and now special assistant to the president, last week. And Francis Collins, who's a practicing evangelical Christian, by the way, Francis said to me, germline genetic engineering, where it's a permanent thing, goes from generation to generation, is a red line, should not happen. And that's his categorical perspective on it. So interestingly, JK called me, he emailed me and said, can we talk? We had a series of long conversations afterwards and on the telephone. And at one point he mentioned an email he got, which he then sent to me. And I don't know if you can read this, but basically what it says, it's from a fertility clinic in Dubai. And it says, congratulations on your recent achievement of the first gene editing baby. And can you give us a course in how to do it? That's essentially what it says. You can see that we now have a tragedy of offshore stem cell centers. I mean, people's lives are being ruined in those places. And can you imagine the seductiveness of an offshore gene editing? They promise not just eliminating disease traits in the lineage, but improving their babies. And a lot of people are vulnerable to a lot of stuff. So I want to show you a video, and here we're going to need to get closer to the I'll just move this back. I want to show you this, take the time to show you this, because this is very important for us to understand. And I'm sure you understand this probably better than I do already, but this is very seductive stuff. The promise of science, the promise of overcoming suffering. And I tell my students, be careful. Suffering in this pluralistic society where we can barely agree about anything. Most of us do agree we should transcend suffering if we can intervene and stop suffering. But the seduction of this idea is that it's the biggest and only moral principle. Yes, it's right to intervene in suffering. We do so in a moral way, but framed with a set of other moral principles, most fundamentally of which is the defense of human life, the dignity of human life. So I want to play you this. This is a political ad that was part of the 2006 congressional elections. Hold on, where's this going? Yeah. And if you think our politics is bad today, I'll be interested to see what you think of this. Come on, work. Oops, hold on, that's not the right one. Hold on. I'm playing the wrong thing here. This is, I'm going to want to play this for a minute too. This is, this isn't working. I wonder why. So what this is I forgot my sequence here. This is an ad from a fertility clinic in New York City run by a Chinese entrepreneur who wanted to set up a very successful fertility clinic for IVF and then move it on toward deeper interventions. And I found out later he was actually working with the Chinese scientist JK. Once he succeeded in his thing, he had the next ambition was to set up a whole series of clinics and make a fortune. And well, you don't need to even hear it. It's basically advertising with a lot of slick photography and a lot of high-tech, stylish, the kind of stuff that plays on people's ambitions and appetites. And it's called Mixed Wisdom and the New York Hope Fertility Clinic. And, all the cachet of American technology and Chinese aspirations to have perfect children. That's what that's about. Let's forget that one, but I do want to play this other one. So we gotta get the sound. I'll let them work on that while I talk to you a little bit about the other sides of this. Do you think you can get it? Okay. So you'll remember we went through the so-called stem cell wars, and there was a pretty bitter controversy at the time. My own alumni magazine had pitted it as science against religion, which is ridiculous, but they didn't mind putting that on the cover. Is it working, you think? Okay. Oh yeah, sure. Okay. So you probably all know that well, here, you'll appreciate this quote. You can read those yourselves. In thy light shall we see the light. Our country has something called the Dickey-Wicker Amendment, which means that we're not allowed to use federal funds for anything that destroys an embryo. But the fight was very deep. And I took this quote off of a advocacy stem cell site for medical use of stem cells, but I thought it was equally applicable to the pro-life position. We fight because lives can't wait. Not just lives to be cured, but lives that are developing. So, I mean, admittedly, it's a difficult controversy for most people to grasp. That's an 8-cell human embryo on the sharp tip of a pin. But if you stop to ponder it, you know that that's the beginning of this great cycle of development that ends up starting with conception, cell divisions, early embryonic development, fetal development, and ends up with a little baby. How mysterious and fantastic is that? But on the other hand, there's lots of sick people and lots of tragedy in life, and that was the compelling argument made for embryonic stem cell research. And I gotta see if I can make this work. I gotta show you this. It's so amazing. So let's see if this works. No, let's. You want to put the microphone right next to it?
I have Alzheimer's.
Okay, hold on.
I won't recognize my husband or my kids.
Okay.
Next week, my mommy and dad here gonna find.
Next summer, I'm going on a camping trip with my friends. On my way home, I'll be in a car accident and I'll be paralyzed for the rest of my life.
In 20 years, I'll have Alzheimer's. I won't recognize my husband or my kids.
Next week, my mommy and dad here gonna find out that I have diabetes.
This is my congressman.
Congressman John Sherwood.
He voted against federal funding for stem cell research.
Is he a doctor?
Is he a scientist?
Why did Congressman Sherwood bet my life that he knows best?
Help me.
Help me.
Who knows? Maybe I'm your mother.
Maybe I'm your grandson.
Maybe I'm your little girl.
How do you know I'm not you?
Stem cell research could save lives. Maybe yours or your family's. Someone you love. Only Congressman Sherwood said no.
How come he thinks he gets to decide who lives and who dies? Who is he?
Majority Action is responsible for the content of this advertising.
So I think you can see what I mean. I think that's a very violent advertisement. And I think in many ways it makes our current problem seem less violent. But because it goes to the very crux of life against flourishing. And that's not a proper division. And I want to say that I live in a medical center. My whole professional life is involved with what I consider to be very, very dedicated and very good people. And even though that they generally come down on the side of the use of human embryos in research, I don't consider them bad people. I think they just haven't thought it through carefully enough. That's my opinion. But they're very dedicated. And they're trying to do good in their lives. So I'm not critical at that level. I just think that we need to collectively as a society think more carefully about what we're doing, which means that we need to pay attention to this ancient Chinese saying that those who choose the beginnings of a road also choose its destination. So most people, it seems, can't relate very well to the inviolability of a very human embryo, or at least there are a great many people. But as it gets older and more developed, more and more people say, no, we shouldn't do that. We shouldn't intervene. But it's the very logic of justification from the beginning that's the problem. It's not a slippery slope. It's a cliff. Once you step off that notion that you can use a human life as raw materials instrumentally for scientific experimentation, you've gone to the bottom already, I think, because you've given up on the notion of a continuity of life's development, and you're willing to use one trajectory of human development for the good of others that have advanced further along. And so that brings us, and I'll just end on these very quick questions. 4 fundamental questions. Will we now endorse the use of human embryos for a wide range of studies of infertility and early development? Well, there are many, many things we could do with those, and I mentioned some of them earlier. And then beyond what, you remember the original argument was over leftover, so-called leftover embryonic frozen embryos and IVF. Well, here's the question, should we create human embryos just specifically for their use? And that you could make the argument we should, because we could genetically alter them. And that, by the way, is the guy who made the first gene-altered mouse. That's Rudolf Jenisch I mentioned earlier. And I also quoted George Daly. Some questions about early human development can only be addressed studying human embryos. Well, even if that's true, does that justify it? Third question, how many embryos is it okay to use in research? Because, some people say, well, a few embryos, that's okay, because just think of all the good you could come from it. That's the kind of utilitarian calculus, but some people think that way, but it won't be just a few. There's thousands of experiments you could do with human embryos. You might even be able to create vast quantities, vats of human embryos, and distill out certain substances that are hard to get. So you could theoretically have millions of human embryos in the mix. Who knows? Right now, the bottleneck is getting oocytes, ovum, the eggs of a female, to join with the sperm. But as I'll show you in a minute, that will be transcended. So how many embryos would we use? Well, one Japanese scientist made 581 copies of a cloned mouse. And you see, the reason to do that is so you get standardized models, and you can give a certain dose of a drug to one embryo and see what happens, and a little more to the next embryo, and you can compare. So that would be very useful too. And by the way, we now can make human cloned embryos. We haven't been able to gestate them, but that may be just a technical problem. So the weak link here is the number of oocytes or eggs to produce these. And back in the stem cell wars, one of the very prominent scientists said that it would be possible to make, since embryonic stem cells can make any kind of cell, possible to make eggs. And because they're difficult to get, and embryos could be grown in culture like any other cell line, and then this problem would disappear. Become like any other cell line, they'd become objects, would be used as objects. We'll probably go through the same agonizing we do with IVF. It could be terrible to begin with, but then it'll become a fact of life. Maybe 20 to 30 years from now, we'll read in the newspaper that someone made 20,000 embryos and studied their development, and we'll decide it's okay. Well, 20,000 is a minimum. It could be many, many more than that. And then, of course, the final question that's a really wrenching question for everybody is, if we do this, how far will we go with it? Can we allow research more than 14 days? As you may know, 14 days is not a rule, nothing to do with the United States policy, except it's a kind of convention. In the UK, it's the legal limit. In China, that's the limit they're operating on, but they don't really believe it matters. And I've been told that by their chief ethicist, by the way. And so 14 days, they made a whole bunch of arguments for 14 days, but they weren't good arguments. And now the International Society for Stem Cell Research, just a year ago, said we need to abandon that 14-day limitation because there's so many good things we can do beyond 14 days. And they put no upper limit on it. So where will this go? Well, in theory, if there were no moral issues, you'd want to study human development as long as possible, right up into fetal development and maybe to birth. And what risks and what would you do? You produce something that's very dysfunctional? Do you just kill it, or what do you do? Well, we need to find alternative ways to study this, because there's no way we're not going to study developmental biology. Either we're going to find moral ways to do it, or we're going to end up being swamped by nations and cultures that allow it, and they'll take over the whole trajectory of science. And I'm happy to say there are some moral ways to do this. I mentioned human-animal chimeras as having, we have to be very careful, but there's some potential there for studying developmental biology. But more exciting is what's called organoids. And they're organ-like things, but they're not in an embryo. And they're no more an organism than just a kidney or a liver is, that we freely take out from living donors. We take kidneys, then we take liver tissue, transplant them into another human. So organoids are partial trajectories of development. And organoids are fantastic things. Some of the major work has been done at Stanford. And I know people who are doing this, and it's phenomenal. Turns out that one of the most amazing things in biology is that stem cells, when they produce, they start out as a simple cell, they differentiate, and then the A goes to B and C, and then B and C interact with each other to produce D, E, and F. And that produces the self-organizing organ development, and the whole embryo, of course, integrated. So it turns out stem cells are very self-organizing entities, and they've been able to produce a whole range of tissues, kidneys, hearts, and even brains that many of the same naturally observed trajectories that we see in where we're able to study the fetal development due to either abortion or tragic loss of pregnancy. So it turns out that you can take these organoids and you can make assemblyoids, which means joining their parts together, and they integrate and function together. And just about 3 weeks ago, an article came out, and this is post-gastrulation. It's about the 12, 14-day stage in human development. They made embryos out of taking embryonic stem cells of a very primitive type. These were cells produced by a method that did not involve an embryo, called induced pluripotent stem cells, iPSCs. Some of you will know what I'm talking about. This method does not involve the destruction of embryo. It takes a skin cell, turns it back into a pluripotent or embryonic-like stage. Then they put some chemicals around it and give it the conditions for it to develop. And they could coax these up to form the placental type stem cells and the early embryonic type stem cells of one part of the embryo, mix them together, and lo and behold, they join forces, they integrate, they communicate, and produce a gastrulation embryo that shows essentially all the same patterns of development as a natural human embryo. These have been carried up to the stage, I'm not sure now what it's at, but 10 days ago, it was 20 days. And the scientific researcher said he was going to take it to 26 days. So we now seem to have what appears to be a way to make human embryos without sperm and egg, and therefore no barrier to creating an endless number of them and studying them. Now, are these human embryos? Nobody quite knows how far they would develop, whether they have the developmental potential that characterizes a human embryo, but you can see that's a tremendously important question. And it's, I won't bother you to read this, but this is really amazing stuff. On another positive front, just last week, my colleague at Stanford, Sergio Pasca, who comes to my classes, a very, very amazing science, he produced an organoid of human brain tissue implanted into a mouse. You probably read this. There's a headline in all over the news, the Drudge Report, and all the conservative and more liberal media all covered this. He produced this entity, this cerebral organoid, implanted it into a newborn mouse, and it integrated. And the organoid actually sent signals to the mouse that changed its behavior. So I think at this stage, I think his work is moral, but it needs a very, very careful analysis for potential problems. And once you get beyond 14 days, many scientists are already uncomfortable with it because they had drawn their marker at 14 days. But my own colleague at Stanford, who's a law professor, said, oh, we didn't need 14 days to be forever. We think, someday we'll change our view on that based on neuroscience. Well, what's in neuroscience is going to tell you that it's a human being? And I think he's going after something like consciousness or some sense of self-awareness in the womb or pain. I don't know what, but you can see that immediately opens up. I think there's going to be a push, if not in the United States, at least somewhere in the world, to allow operation to the production of human embryos, altered and studied, but developing not just 20 days, 28 days, but to the level of implantation. We're developing artificial wombs to develop these things in. In fact, I have a bet with one of my colleagues at Stanford, a very distinguished embryologist, who my bet, I'm actually on the pessimistic side. Well, it's hard to say what's the pessimistic. I'm on the side that says this won't happen. She thinks within 10 years, we'll have a fully born baby gestated completely in an artificial environment. And she knows more about it than I do, but I think I know a little more about medicine, how complicated it is. All the best laid plans of physicians don't turn out, do they? But nonetheless, it's coming. Sooner or later, we're going to have to contend with the Brave New World scenario of baby production. And where will this go? Well, some people say you're not a human being. This is the head of the Biomedical Ethics Center said you're not a human being until after 26 weeks. That's ridiculous. We have babies in the ICU, NICU, that are less than 26 weeks. And there are philosophy professors said, oh, early human embryos are like plants. They don't really count. They're living things, but there's nothing moral about them. Ideas in different countries, and Chinese ideas that you're not a person until after birth. I gave a talk in Beijing, and the chief bioethicist said, we do the 14-day rule, but we don't believe you're a person until after you're born. And afterwards, a philosophy professor took me aside very quietly where nobody could see him, and he said, no, that's not what Confucius says. But that's the modern Chinese interpretation of what Confucius says because it comports with what they want to do scientifically. So you can see worldwide huge challenges. Now, why would anybody want to do this? I'm going to skip this. This is the justification in the UK for doing 14 days, which now is being transcended by the revision of the policy maneuvers. And here's the thing I want to tell you is one of the great dangers is that conceivably scientists will be able to form human embryos, take them to a certain stage and harvest out their cells, tissues, or organs for therapeutic use, including whole organs for transplantation. I don't know if this will actually work, but there are scientists trying it, and they're doing it with aborted fetal parts. So, for example, in the United States, there are something like 20 people a day die of kidney disease because there are not enough kidneys. So if you could grow kidneys in a human embryo and harvest out the early kidney primordia, maybe that would solve the problem. before you could actually produce them in an isolated factory and just produce. I mean, as strange as it sounds that we live next door to the kidney factory, it's probably not immoral. It's just a little kind of ghoulish. But growing them in embryos raised a whole nother question. I want to just tell you a couple more things before I stop because they're kind of interesting. So 30 years ago, I was friends with a guy at Stanford who was working both at Stanford Lab and in a private company called Systemics that was south of Stanford in the Silicon Valley area. And I wanted to see what he was up to and what they were doing at Systemics. So my friend took me for a walk through the lab. And as we were going through the lab, he reached down on the bench and he picked up a little test tube and said, "Look in there." And I looked down in the test tube, and there at the bottom was a little tiny 3/8 of an inch hand, a little human hand. And I said to him, "Where did you get that? This was so perfect." And he said, "Well, we took a 4-week-old aborted fetus," because they're using aborted fetal tissues, and we snipped off just a little tiny bud, and I'll show you what that looks like. This little tiny bud off the trunk of the fetus. First, it looks like a cylinder, and then these little buds come. And we snipped off this little bud, and we put it in the space of the abdomen of a mouse. And it was a kind of mouse that would not reject it for immune response. And we let it grow, and 3 weeks later, we opened it up, and lo and behold, that little clump of cells had turned into a hand. And this is the way it works in natural development. This little bud sends out these little shoots that form the arm and the hand. All the information is there. First, it starts out as like a little paddle with a webbing, and then those cells die, and you get the little fingers. And then in a normal development, it goes on to form the little hand of the fetus. And I looked down at that little hand in the test tube, and I thought to myself, wow, this is fantastic. Cells are self-organizing. We may someday be able to provide hands and limbs for amputees and who knows what other organs. But then it took me about a minute before I realized, yeah, but that was going to be somebody's little hand. And I thought of the little hand that lays across. I have a bunch of kids, and I watch them nursing, and the little hand that lays across their mother's breast while they're breastfeeding, and little eyes look up at you. And I thought, wow, this was going to be somebody's hand. But now it's in a test tube. And it really raised for me the magnitude of the meaning of what we're up to. It's very positive in its possibilities if we can do it morally, but it's horrifying if we don't. And there's actually a scientist there are actually scientists studying this in Israel and Japan, and way back when I was in the President's Council. And there's an ethicist who's also a physician, runs a ethics think tank, bioethics think tank at Oxford University, a distinguished university. He maintains it's morally required that we employ cloning to produce embryos or fetuses for the sake of providing cells, tissues, and even organs for therapy, followed by abortion. So you can see where this could go. A very great horror. And as I said, there's a private company, I think they've gone out of business now, but they were trying to use fetal parts to do this. So where will this go? Well, this guy thinks that it's women's right to not only, if they can have right to have an abortion, then why not a right to use their pregnancy for some kind of a economic benefit to them? And he goes on to say that many women would like to use the proceeds of sales of their fetuses to finance college education to help raise their children. What's he talking about? Choice need not end at the removal of the fetus. It's what you do with it afterwards. Someday, if we are fortunate, he says, scientific research may make possible farms of artificial wombs, breeding fetuses for their organs. This is a bioethicist. Babies in bottles. Well, where are we going? Artificial endometrium. It's coming slow but surely. So this is a quote from Leo Alexander, who was a very, very thoughtful physician who witnessed the Nazi trials, and he wrote the Nuremberg Code. And the operative part of this is that the Nazis' error started from small beginnings. The beginnings were merely a subtle shift in emphasis in the basic attitude of the physicians. It started with the acceptance of the attitude basic in the euthanasia movement, that there is such a thing as a life not worthy to be lived. Amazing thing. We all know where that went. Nobel laureate discovered the double helix, Francis Crick, has said that no newborn infant should be declared human until it's passed certain tests. And if it fails them, it forfeits the right to live. But another scientist, Erwin Chagaff, very, very smart guy, warned us what was coming. He made key discoveries in 20th century genetics. And Chagaff said, science is now the craft of the manipulation, substitution, and deflection of the forces of nature. What I see coming is a gigantic slaughterhouse, an Auschwitz in which valuable enzymes, hormones, and so on will be extracted instead of gold teeth. What is madness? Wrote Voltaire. To have erroneous perceptions and to reason correctly from them. We're at a crucial moment, crucial juncture in human history, which could lead to widely contrasting futures. Great powers are coming into our hands. Those hands, the little hand of the baby in the test tube, the little hand in the poster I showed you operating on itself, and that hand touched by the Spirit of God, that hand which is so powerful, so amazing within the cosmos and its comprehension and control. And in this, he showed me something so small, no bigger than a hazelnut, lying in the palm of my hand, as it seemed to me, and it was as round as a ball. I looked at it with the eye of my understanding and thought, what can this be? I was amazed that it could, for I thought that because of its littleness, it would suddenly have fallen into nothing. And I was answered in my understanding. It lasts and always will, because God loves it. And thus, everything has being through the love of God. We need to be like St. Francis. We need to answer all of our problems with more love, not less love. Thank you very much.